Swayam Prabha. Digital India. Educated India.
So, maternal effect means it is a product that is there already in the oocyte, and it will be there in the oocyte if the mother made it. Okay. And if it does not matter whether oocyte genome has it or not. So, it is going to function normally. So, usually such gene products function in the early embryo. Okay. So, you do not expect the maternal products to hang around late in adulthood. So, therefore, you always see the phenotype in 2 generations later. So, usually in the grandchildren generation. So, what we are right now going to look at is, so far what we have done is we first looked at the oocyte, how oocyte is getting the axis, anterior posterior axis formation there. Then we saw the dorsoventral asymmetry there. So, now we are going to look in the embryo. Okay. So, in the embryo we are going to look at the pattern formation. So, see, initially you have a pattern established already of the asymmetric localization of bicoid and Oscar, right, in the oocyte. But that alone is not going to be enough to make a complex organism. So, you need to enlarge that asymmetry, and that asymmetry has to become very localized as well. So, then only you are going to make multiple tissue types and at the right places, therefore, the organs form at the right places. So, you need to enlarge this asymmetry into many more molecules. So, how that plays out has been best understood in the Drosophila embryogenesis, and that is why we are focusing so much attention on it. So, the first asymmetry starts with the maternal factors, okay, meaning these are products of the maternal germline, and they have already come either as protein or mRNA into the new generation, new embryo. Okay. So, how they are going to be asymmetrically localized is the first part, and that is what determines the next set of genes. Okay. So, these are usually mRNA, and they are asymmetrically localized or asymmetrically translated or both, and that is how this initial pattern forms. Okay. And some of them are translational regulators as well as transcriptional activators or inhibitors, like bicoid we saw is a translational inhibitor of caudal, but at the same time it is going to be transcriptional activator of genes that we have not yet discussed. And their asymmetric localization, finally the protein product, is going to turn on the transcription of downstream genes called gap genes. Okay. So, they are called gap genes because their mutations led to large gaps in the embryo that develops. Okay. So, you are going to see that today we will go through all of them. So, you will see examples and what the gap is in terms of the embryo, all that we will see. So, their expression is like this, large patterns. Okay. They are not like gradient in this fashion, but instead you have, see here you see 1, 2, 3 and that repeats again. Okay. So, this larger band like expression in the embryo, and they in turn based on, and they do have overlapping distribution as well. So, these stripes are not showing any overlap, but they do overlap, like for example, this orange color may overlap some part of this purple, and purple may overlap this, and that overlap and exclusively this orange or the exclusively purple, all of that matters in terms of which set of next genes are going to be turned on, and they are called the pair rule genes. They are called so because of these stripes like expression pattern they form, and they usually affect every other segments finally in the larva that forms. So, you understand segments, right? Some of the organisms have segmented body pattern. If you take any insect and look at it, you can clearly see a larger abdomen segment and thoracic segment where you have the legs and the wings, and then you have the head where you have the antenna, etc., right? You can see this in any insect. So, our body is not like that. So, you need to understand that some organisms have segments in their body, and this makes actually 14 segments, okay. And these pair rule genes affect alternative segments, okay. So, their expression pattern forms these very precise sort of stripes, okay. So, you see from this sort of a gradient, now you have gone to this pattern. So, you are only looking at one example, there are multiple pair rule genes, and the pair rule gene products, gap gene products, and even some of the maternal products that are transcription factors all together coordinate to activate next set of genes called segment polarity genes. So, these genes specify, you see the expression pattern here, in each segment this particular one is expressed in the anterior part, okay. Anterior is always to the left, okay. So, these affect a part of segment in every segment in the mutant. So, these are the segment polarity genes, and by this time actually the embryo gets cellularized. By the time segment polarity genes are activated, it gets cellularized. Now, you can have adjacent cells signaling and influencing each other, and that finally leads to activation of what is called homeotic genes. So, today our goal is to go up to segment polarity genes. Homeotic genes we will see next class, okay. So, hopefully we will go up to this. Alright, so start with maternal. So, maternal we are kind of already familiar, bicoid that got to the anterior in the oocyte itself, remember, and now that gets translated and forms product, okay. So, while these are happening, our dorsoventral thing is also simultaneously happening, okay. So, the gherkin having gone to the dorsal anterior, and there it is suppressing pipe in the dorsal follicle cells, while the ventral follicle cells get primed to transduce the signal via the Toll receptor there, and that signal transduction is required only when dorsal protein is formed quite a bit later. So, while that is happening, so parallelly in the embryo this is happening. So, the bicoid mRNA that was localized, now it gets expressed, and then you have this gradient. I told you the gherkin going to the dorsal anterior and inhibiting pipe synthesis in the adjacent follicle cells, that happens during the oocyte stage, but the signal transduction at the ventral and the dorsal getting only into the ventral nuclei by the time the embryo has gone into blastoderm stage, where the nuclear divisions are over and the nuclei are on the cortical location, okay. So, it is happening continuously. So, while that process is happening, similarly you have this anterior posterior axis formation is happening too, okay. So, these you should not disconnect them. So, for convenience sake we are ignoring all that discussion that we had so far. That does not mean that happened in one embryo and one place and time, and this is happening in another place and time. So, while you have that follicle cells being specified ventral dorsal, in the embryo now fertilization happened and the anteriorly localized, similar to the dorsoventral thing, that part happened in oocyte, part happens in the embryo. Similarly, the anterior posterior also part happened in the oocyte. Remember the asymmetric microtubule formation, and as a result a bicoid hitching a ride on a motor protein and going to the anterior, and the Oscar going to the opposite side, right. So, that is already an anterior posterior patterning forming, that is at the oocyte, But what I am saying is now that leads to that needs to be enlarged further into more molecules, therefore you can make more specific structures. You can't end up making only 2 domains, you know, an anterior pole and a posterior pole, and that alone is not enough. You need to make multiple structures, multiple tissue types. So therefore you need to enlarge that. Okay, so you are initiating an asymmetry, but then that asymmetry you have to build upon that asymmetry into more localized asymmetries, right. So that is what now we are seeing. This does not mean the anterior posterior is starting only in the embryo. So this is oval shape, this is proper embryo, this is no longer oocyte. So this does not mean it is only happening now. We already saw the microtubule orientation happening in the oocyte, and as a result bicoid mRNA has gone to the anterior and so on in the oocyte. So now you have the protein produced and the protein forms a gradient. Okay. So the highest concentration of bicoid at the very anterior most activates certain gap genes, you know, that we are not having it in the next image. So those are required for the anterior most segment developments, and there are structures called acron that forms anterior to the segmented part of the body. So you have a portion, a tail like portion called telson, that is coming in the next slide, so do not worry about the spelling. And similarly anterior to the segmented part of the body is actually the anterior most part of the head of the fly. So that is where actually the brain resides, and they are outside of the segmented part, those two, and that structure formation requires the highest concentration of bicoid and other factors as well, we will see later. And in the not so extreme but still anterior portion is where it is going to suppress the caudal expression, and in turn it allows the hunchback expression to happen there. So it is a transcription factor as well, and it is going to activate hunchback. So hunchback activation does not require high concentration of bicoid. Okay, so there are other names, I do not know whether those names are there in the slides, but otherwise I will tell you what I remember. They are all funny names. There is something called empty spiracles, orthodentical. So those kind of genes are the anterior most, and their activation requires bicoid. And then you need a slightly lower concentration of bicoid for activating hunchback. So these are all determined by primarily the affinity between the promoter agent, rather enhancers, and these transcription factors. And so the bicoid is at the anterior. So these are, we are still basically what we are doing is the same cartoon, we are seeing actual examples. We have not actually still gone into what is the maternal gradient, what is the gap gene, etc. So these are still simply examples, we will get into the details in a minute, I guess. So essentially instead of seeing this cartoon, now you are seeing an actual example. Okay, so you are not in this particular embryo, this does not have the immunostaining or in situ hybridization done for the posterior localized protein or mRNA. And this is an example of 3 different gap gene expression patterns. So you can see hunchback expression here, and you have the cripple, that is another gap genes, and you see this overlap, that is why you have the orange. So this overlap is not shown in the cartoon, okay, but here you do see. So these few cells, these band of, see you can see the nuclei here. So this is a surface view, so therefore you are able to see the nuclei here, okay, and you can see these nuclei have a unique pattern of proteins being produced there. So they are making both hunchback and cripple, while here they do not make hunchback and here they do not make cripple. Okay, so this is gap gene. So do not worry about this hunchback expression. This is a specific domain of hunchback turned on by another gene that really does not fit in the overall scheme that we are discussing. Okay, this is an example of pair rule gene. So the Japanese group did, and therefore it is Fushitarazu. Okay, Apparently what that means is fewer segments. Okay. So this is expression pattern of a pair rule gene. Okay, this is no longer cartoon, it is an actual embryo showing actual distribution of a given molecule, Fuchs-Tarazu protein. Okay, this is a late pair rule gene. So there are pair rule genes actually are two groups, one is the early pair rule gene and then a late pair rule genes. Together there are 8 of them, and we will see them in detail. I think there will be a table as well. And then this is an example of segment polarity engrailed. So this is there in, so this area is one segment, and you see this only in the anterior part of the segment. So you can count here, you will find 14 of them. Okay. So these are examples, we will get into each one of them how their pattern is established, that is our today's goal. Alright, it all started with very simple observation in as early as 1975, when Klaus Sanders did a simple experiment. So he did not take Drosophila but a different insect. See here you can see the segments, even the abdomen has segments, can you see that? So these organisms have segments. So here the interesting experiment they did is they simply took an early embryo and tied a thread in the middle of it, actually a ligature in the middle, so that things of the one side anterior can't go to the posterior, posterior can't go to the anterior. Okay. So they kind of tied there in the middle. Then they realized when they do that, anterior made the anterior, posterior made the posterior, but the middle structures did not form. And the later and later they shifted their ligature timing, they found it basically reduced lesser and lesser number of missing segments, middle segments. Okay. So based on this, what Sander proposed was there must be two decision points, one in the anterior and posterior, and they probably instruct the rest of the things in a gradient fashion to make the different structures, the middle segments. Okay. So that is the conclusion he came to. And then he also, so these observations suggested the existence of a morphogenetic gradient. Okay. That is the two centers of morphogen production emanating from the two poles. And they did one more experiment, that is they irradiated the anterior part with UV, so that the nucleic acids, like for example an RNA, if it is there, that is gone, or they injected RNAs to kill RNA, and then they found when they did that the head structure did not form in that embryo. Okay, it formed two tail like structures, okay, indicating that an RNA is probably behind this morphogen gradient, at least from the anterior. So this observation led to Naslin Walhard and her group to ask questions, what are these morphogens, how are they forming the gradient, how these structures are made, and how those asymmetries are stabilized, and so on, multiple questions. And then the approach they took is mutagenize and look for mutants in a very systematic way to see what structures are missing in what and make sense out of them. And answers to those questions is what the summary we just saw, the maternal effect, gap gene, pair rule, and so on. So now we are going into the details of each of that. The first one is maternal effect genes. Okay. So we already kind of learnt earlier bicoid, in the differential gene expression topic we learnt about bicoid being in the anterior, and her experiments of introducing bicoid mRNA into the mutant, or no, to start with the anterior cytoplasm she injected into bicoid mutant embryos anterior or middle or posterior and so on, right. So here the summary is the anterior has the bicoid and the posterior is nanos. Okay. So I will tell you a little bit about nanos. So we let us spend enough sufficient time to understand this, because we do not have multiple slides explaining each of the steps. So nanos mRNA is produced by the nerve cells and they are deposited into the embryo. So, nerve cells, if you remember, they are in this part to start with, they are anterior to this structure. So, they all enter in the anterior, and the nanos mRNA that sort of diffuses, it is not hitching a ride on those motors zooming on those microtubules. Instead, it diffuses freely in the cytoplasm, and whatever goes to the posterior, who is already there in the posterior? Oscar. Okay. So, Oscar protein is by then produced, and that Oscar protein can bind and hold nanos mRNA there. Not only that, an Oscar can dislodge some of the inhibitors, translation inhibitors, and allow nanos translation in the posterior. And the nanos in the bulk cytoplasm is bound by protein like SMOG and another one called CUP. These names are not going to come, so I am going to, but all of these are there in the book, SMOG and CUP. Okay. So, these proteins bind to nanos 3' UTR and prevent its translation in the bulk cytoplasm. So, the summary is nanos is produced only here, bicoid is produced only here, and they form this concentration gradient. Okay. And this is happening, you see this gradient is existing in the syncytium, right. You see the nuclei nicely arranged in the cortical region alone. Okay, so, only here. So, upon cellularization as a downstream product, you get all these activated. Okay, so, that we do not need this portion for now. And so, this is what initially happens. So, this is the same experiment we have already discussed. This is just that it is having more specific labels like acron, telson is marked here, and head, thoracic and abdominal segments are marked, and this is the structure wild type forms by having bicoid here. Now, if you do not have it, it forms abdomen, telson, indicating that the anterior structures like this acron, head and thoracic, these formation required bicoid. Okay. And this we sort of know, like introducing bicoid mRNA instead of the anterior cytoplasm in this experiment, it is more little bit more sophistication. You now know what is the molecule and you are introducing that specific molecule. So, when you introduce bicoid mRNA in the anterior, then you get the normal wild type, right. So, now you put bicoid in the center, then you get head there, you do not get acron, but you get head, thoracic and abdomen, behaving more like the proper definition of obeying the definition of morphogen gradient here, right. And then in the wild type you put in the posterior, okay, wild type it is having the bicoid there, it is going to make the normal this embryo. Now you put bicoid here and you end up making an acron there and then a head and thoracic. So, double headed insect you can make. So, actually many mythological characters can be made if you understand the developmental program and the molecules. Okay. Double headed organisms are known well, anterior, posterior or head to tail axis duplication leads to double headed tadpoles, for example. So, this shows an in situ hybridization for bicoid mRNA and then you see this gradient here. Okay. And this is bicoid protein and this forms a shallow gradient that people believe is because some amount of RNA that is here, even they get in and they are already poised for translation and therefore, in a very short time you get this gradient of bicoid formed, and this is the mutant and this is the wild type quantitation of that protein signal. So, this is how the surface cuticle looks like of wild type and bicoid mutant, which is missing some of the head structures. All right. So I did not tell you what the bicoid does, but you kind of already had this information earlier. So bicoid gradient in this portion, here it suppresses caudal and it activates hunchback transcription in those nuclei where it is already there. Remember, it is a transcription factor as well. So the caudal is by translational suppression, but hunchback activation is by activating from the zygotic genome. So you distinguish zygotic genome from maternal genome. Okay. So bicoid mRNA is product of maternal genome. Okay. So if the mother had even one allele of wild type bicoid, then you have the bicoid protein like this in this embryo. So the embryo has its own genome, it got a, it is formed by two gametes, each gamete had one copy, one allele of a gene, right. So if both the alleles are mutant, does not matter, it is going to form that. Caudal similarly, but this hunchback production, hunchback is both actually, that is maternal as well as zygotic and both function. So the zygotic hunchback activation, so most of the gap genes are zygotic, okay, so that is why we do not call them maternal. Hunchback is kind of exception, it is both maternal and zygotic. So from the zygotic genome you activate the transcription of that. So remember this again, people use these terms quite often and they have very specific meaning. When you say zygotic gene activation or zygotic genome, they mean the genome of this new generation, so that new embryo. Maternal means from the maternal genome you would have had the products sitting in the oocyte cytoplasm, and that matters in this current generation of the embryo development, and that is why we need to consider that, otherwise you can simply ignore it. So higher concentrations, yeah, here are those names. Higher concentrations of bicoid at the very extreme anterior, this is the name I forgot, button head, empty spiracles and orthodenticle. These are the gap genes that are activated by highest concentration of bicoid at the anterior most, and slightly less concentration is good enough to activate hunchback. And at the posterior, caudal again is a transcription factor, and that is going to activate posterior gap genes, and they are NIRPS and giant. Okay, these two are activated. You are going to find later that giant is actually activated by bicoid as well. So giant is going to be produced in two places. So this is the summary of the anterior and posterior set up by the maternal effect products gradients. Okay. So we haven't seen one part of it, that is we put the nanos in the posterior. So what is nanos doing at the posterior, right? So nanos actually binds to the hunchback mRNA 3' UTR and along with pamelio. So both these proteins bind to hunchback 3' UTR and suppress hunchback translation in the posterior. Remember, hunchback comes as a maternal product as well. So that maternal product that is distributed throughout the cytoplasm needs to be inhibited in the posterior. Therefore, it is not going to activate this thoracic segment related genes there, so that it allows abdomen development. Okay. So that is going to come next. So this is caudal, you see it is a mirror image of bicoid, again it is in the nuclei. So to remind you again what is happening developmentally, these are syncytium not cellularized. Okay. So you have a lot of nuclei in the common cytoplasm. Only thing is the nuclei are on the cortex. So you are seeing the surface, therefore you are readily seeing the cortex, right. If this is the embryo, so cortex would be towards the surface on all sides, right, all 360 degrees. So if I am going to focus on the very top, I am going to see like this. So now enter the another posterior, the posterior molecule, which is nanos. So nanos mRNA we already understood how it gets here, right. Oscar binds and stabilizes it there. Here while what is in the bulk cytoplasm, it is actually going to be not translated because of the smog binding, and also the smog and cup, they are going to remove the poly A tail, and as a result nanos mRNA gets degraded as well, it does not get protected here. So, due to that you see nanos RNA primarily here, and that some amount is left here and that is not readily visible here. So, you actually have a gradient, but a steep gradient mostly here and very little here, and that very little is translationally inhibited, that is not going to be translated. And as a end result in the protein you see this gradient, a shallow gradient going from here to this. Okay. And this nanos protein produced here in collaboration with Pamilio bind to hunchback mRNA and suppress the translation of hunchback, and that is going to prevent the anterior structures forming here, and that allows abdominal structures to form. So, this is a summary of that, the cartoon, and this gives you hierarchy the same way we saw for the dorsal. So, you have hunchback mRNA, yeah, this is one thing I did not tell, so that's why it is good to have the slides. So, hunchback, while the protein is going to be produced or not produced due to what the bicoid and nanos have to say about hunchback translation, mRNA itself is distributed all over. Similarly, caudal mRNA is there all over, but caudal mRNA would be not produced converted into protein here, and hunchback will not be produced into protein here. Okay. So, this is the RNA gradient, and this is the bicoid RNA, this is the nanos RNA, and later, so by this is oocyte mRNAs, right, by early cleavage embryo. So, what you have is here, because you do not have nanos, hunchback gets translated and you make protein here, and here nanos is going to suppress, and as a result hunchback is not going to be produced, and here you do not have bicoid, and as a result caudal is going to be translated. So, you get these from these steep gradients you go to this shallow gradients of proteins. Okay, so this is the gradient, and this is the sequence of events. Bicoid, so we did not talk about this, these things promote its translation to make bicoid protein, that is going to suppress caudal translation. Okay, that is the anterior. In the posterior, nanos helped by Oscar primarily relieving the translational suppression, it becomes nanos protein, along with Pamilio and this P55, it is going to suppress hunchback mRNA getting translated into hunchback protein. This is what happens the posterior. Alright, so we are done with the maternal part, and before getting into the gap genes we need to take care of these extremities. Okay, so how those extremities are formed, and that is by this RTK signaling. Okay, so there is a protein called torso. Okay, so this torso is responsible and for transducing a signal, and that signal is torso like, and this torso binding to torso and activating MAP kinase cascade is what leads to the torso signal. This does not happen all through, this is happening in the follicle cells on both sides, it does not happen everywhere, that is primarily because only these follicles produce this torso like protein, while the torso itself is expressed all around the oocyte membrane. Okay, so the receptor, this RTK receptor is there all through the membrane, but its ligand comes only from the two ends. Okay. So, as a result you have that signaling there, and what is the consequence of that signaling? This protein Gaucho is going to be suppressed translationally, and therefore it forms this sort of a reciprocal gradient, reciprocal to the torso signaling, and this Gaucho, if it is there, it is going to not allow huckabine and tailless production. Okay. So, those two protein production is required for these extremities to form. If those two are produced and you also have bicoid, you make the head. You do not have the bicoid, you make the tail. Okay. Very interesting consequences of these asymmetries, right. And that is how this torso signaling. So, you are seeing an actual example of a MAP kinase pathway here in development. So, that is how you have actually the same genes, Huckabee and tailless, produced by the same gradient. You just can't explain any other asymmetry to make this produce head and this to produce the tail region, and that asymmetry is provided by the bicoid being present here. So, this is how the extremities are taken care to make the acron and the tails and structures to form, right. So, next we are going to look at the gap genes. So, we are done with the maternal effect proteins forming the anterior posterior embryonic patterning. Here patterning means the pattern of distribution pattern of the molecules, and we saw the extremities, and now we are going to look at the consequences of these gradient setup. And one of them is we are going to look at gap gene crupple. I do not know how it is pronounced with the two dots over the U, but I am going to say crupple. Okay. So, this one, this is a cartoon showing its approximate region of expression and what happens in the mutant. So, it is somewhere here, and in the segmented region like this they call as germ band. So, it is in these many segments. Okay, it is a broader body region, and then in larval stage it is these structures, and if this protein is not there, then these are going to miss in the mutant, and that is exactly what misses. So, what is missing directly corresponds to where this protein is produced. Okay. So, this is pair rule gene Fuji Tarazu, and again you see it is in the very early syncytial embryo you have this expression pattern, and when you have the segmented larval stage or sorry late embryo, you are going to have focus on this anterior of one and posterior of the adjacent, right, anterior of one and posterior of the segment anterior to the first one, right. So, it is expressed in that region. So, this is called para segment, and we are running out of time. Para segments I will explain in the next class. Okay. So, what is segment and what is para segment, but right now all you need to focus is, therefore, in the larval stage this is where the expression pattern will be, and as you would predict in the mutant anterior part of this segment and posterior part of this segment will be fused and be present. Okay. So, one segment's posterior part, another segment's anterior part will be missing. So, wherever it is expressed, that is what is going to be missing. Okay. And segment polarity gene on the other hand, it is in more stripes, these are actually 14, and they are on depending on this engrailed is on the it is in the posterior of each of these segments you are seeing here, and similarly you see in the larval stage, and then only part of every segment is going to miss in the mutant.